Thank you very much. We will now go on directly to the remote presentation by Dr. Diamond.
Great. Thank you for the opportunity to talk, and I really apologize for not being able to get there. As Tom Moon alluded to, sometimes the airplanes and weather do not cooperate. However, I will try to make some points which I think are important for understanding Zika virus disease pathogenesis. Can I have the next slide, please? So, as mentioned by other speakers, Zika virus is a flavivirus, and that means it has the same canonical RNA structure with 10 genes, which are 3 of which are structural and 7 non-structural, as you can see on the top. The flaviviruses are related more distantly to other members of the Flaviviridae family, and this includes hepatitis C virus. And I'll mention hepatitis C virus a little bit later, that's why I raised it. These are positive polarity RNA viruses. So the flaviviruses, most of them are mosquito or tick transmitted. And in this talk, I'll review some of the disease syndromes that they actually cause and where we think Zika fits into this. And so the flaviviruses, for the ones that cause epidemic disease in humans, tend to cause either encephalitis, shock syndrome, or liver failure. And the 5 major ones now that we think of really are Zika virus, obviously the discussion today, but dengue virus is the one that actually causes most disease, where there's 390 million infections per year. And then West Nile virus, as we know, which is endemic in the United States, Japanese encephalitis virus, and yellow fever virus. And I should also say tick-borne encephalitis viruses. And these are different in the context of what types of diseases they cause and the particular mosquitoes and also the regions that they do this throughout the globe. Next slide. So this is a dendrogram of where Zika virus stands, and this was alluded to earlier in the talk by Dr. Rosenberg, but also by others, about the relatedness to this. And you can see Zika virus actually is very close to another virus that we don't know too much about, the Spondwelli virus. However, as Richard alluded to in the last talk, it has a significant amino acid similarity and homology to dengue virus, although it falls in its own virus clade. It is more distantly related to the encephalitic flaviviruses, including West Nile, Japanese, and St. Louis, and also to the tick-borne viruses. And one of the other interesting features about Zika, of course, is that it's a mosquito-transmitted virus. As it was alluded to, the Aedes seems to be the dominant mosquitoes, and also there is certainly in the old world evidence of non-human primates as being a sylvatic reservoir. Next slide. So one of the questions I wanted to address is the pathogenesis of Zika a different, unique, how does it relate to the other flavivirus infections? And so I'm just going to review the 4 major virus families now in terms of what the diseases they cause in terms of where Zika stands. And so the encephalitic flaviviruses, which include West Nile, Japanese, tick-borne, as well as St. Louis encephalitis virus. These viruses effectively replicate locally wherever the mosquito bites. They spread to the draining lymph nodes, and then there's a viremia period, and then ultimately they spread to the brain and spinal cord, and they infect neurons. And it's this direct infection of neurons which can lead to injury directly, but also by bystander injury, and this leads to cell death in the peripheral as well as central nervous system neurons. And this has a clinical outcome of encephalitis, meningitis, but also, as we saw in the United States during the early outbreak of West Nile virus around 2000, of acute flaccid paralysis. So these are really what we call encephalitic flaviviruses that target neurons. And then there are the hepatotropic flaviviruses, which yellow fever is the primary example, which can infect liver cells and cause injury, but also can cause injury in the kidney. And you can get a hepatorenal syndrome. And this can lead to acute liver and kidney failure, and this can be very devastating. Although, fortunately, we've had a vaccine for yellow fever, which has been used in hundreds of millions of people effectively since the 1930s and 40s. And then there's the hemorrhagic fever and shock syndrome, which includes dengue virus, which infects myeloid cells, monocytes, macrophages, and dendritic cells, and can produce high levels of pro-inflammatory cytokines. And this, along with some interesting factors about the virus biology, and particularly the NS1 protein, can lead to a shock syndrome or hemodynamic instability, as well as bleeding syndrome as well. Where Zika fits into this, we still are really at the beginning of understanding it. Why is there microcephaly? What about Guillain-Barré syndrome? And is there evidence that it also is a neurotropic virus? Next slide. So I just want to review briefly some of the clinical scenarios that we see for the different viruses. So this is West Nile virus, and its major effect is the mosquito will inoculate the virus into the skin. It replicates locally, and then it causes viremia. And then the key question is whether the virus is able to cross the blood-brain barrier or cross into the spinal cord. If it doesn't, it remains an acute febrile illness in many people. Some people actually don't even develop any symptoms, and then it gets cleared over time without any sequelae. However, in a subset of individuals, and for West Nile, it's primarily people who have comorbid illnesses or elderly, the virus can cross into the brain and infect neurons, and this is how it becomes an encephalitic virus. Next slide. In contrast, dengue virus has quite a unique pathogenesis where myeloid cells are the primary target, and this leads to the clinical symptoms that you'll see on the left, including the severe vascular symptoms. But really, it's due to something about the unique biology of dengue virus, and that is that there's 4 different serotypes. And the most severe disease of dengue actually occurs not the first time you get it, but actually the second or potentially the third time. And the reason why is it's an immune enhancement disease. And studies over the past several decades have really led to a much greater understanding and resolution of this clinical disease pathogenesis, such that after the first infection, you develop antibodies to the original infection you had. For example, if you had dengue 1, you develop antibodies to dengue 1. These antibodies are able to then protect against dengue 1 pretty much for the life of the individual, although there is some question to that. However, these antibodies on occasion can cross-react to dengue 2, dengue 3, and dengue 4, and if you get a second infection, they cross-react, but they do not neutralize. And so what happens is the virus, you get an antibody-virus complex, and this is able to then facilitate entry into a monocyte or a macrophage or myeloid cell, which expresses Fc receptors, and this leads to higher levels of production of virus, which also leads to higher antigenemia as well as higher cytokines. And it's the combination of the cytokines as well as antigenemia, which we believe then changes the endothelial cell permeability, which leads to a plasma leakage syndrome, and this causes the unique dengue pathogenesis. So in this case, the second infection is usually the one that's worse, although this actually also happens relatively infrequently. Next slide. So what about Zika virus? What do we know about it? Is there animal data that can help us? Well, as I think Scott alluded to quite well in his talk, and Tom Monath as well, we've known about Zika for a long time, but because it did not cause severe clinical syndromes that we were aware of, it just really wasn't studied significantly. While we have studied other flaviviruses, there are still a number of questions. And so what do we know about animal models? Well, Scott alluded to the 1947 to 1950 studies where it was originally isolated. There were 6 rhesus macaques in the towers. One of them became febrile. A blood sample was taken and then injected into mice into the brain, and this led to a number of mice getting sick, and then the virus was packaged. And this generated the original Zika stock, in fact, that is still being used in laboratories today. However, that virus was only found in the brain and the spinal cord, suggesting that Zika virus might have a propensity for neurotropism. However, it was directly injected into the brains of mice. And then the only other study that actually has been performed in mice was in 1976, at least that's published, and that is when another investigator took Zika virus as part of a series of a larger panel of viruses and was able to inject it into suckling mice, and then show that you could also inject 4-week-old mice into the brain. But in this case, mice got sick, and mice died, but there really was no pathologic description. So in the past many decades, we really had a paucity of published studies on Zika virus infection in mice or in monkeys to work with, and so this leads us to a number of unanswered questions that we'll have to address. Next slide. So what are the urgent needs from the standpoint of understanding pathogenesis? We need animal models of infection and disease pathogenesis with circulating strains. Scott alluded to earlier that there may have been changes in the amino acid and nucleotide sequences which can alter the potential not only for transmission in the mosquito population, but also for pathogenicity and virulence. And we really need to understand that using reverse genetic analysis, where we can make mutations into infectious viruses and test hypotheses about why the current outbreak seems to be worse. And then we need mouse models and non-human primate models, and really we're at the beginning because nothing has really been done for many decades. We really want to understand how the virus infects, how it disseminates, how and does it cause Guillain-Barré, and of course establishing maternal transmission models. And in humans, outside of the animal models, we really want to perform longitudinal studies to follow where the virus goes and what's the nature of the injury, perform detailed pathologic analysis, and really answer the question, why is it worse now, and why is it only in some people? Are there genetic factors or particular acquired factors which can lead to more severe disease? Next slide. So I just want to review the two recent publications on Zika virus microcephaly. In the earlier talks, it was alluded to that there's mounting evidence now that Zika virus is associated with microcephaly. And two recent case reports, one in the New England Journal and one in the MMWR just last week, really makes Zika as a likely cause of microcephaly now. In the first paper, there was 2 newborns with microcephaly that died soon after birth and 2 miscarriages. And in 4 out of 4 brains that were harvested from these tissues, they were positive for Zika virus RNA, and only the brains were positive. The genetic sequence of Zika virus was obtained in fragments, and there was viral protein seen. And in the second paper, which was due to a terminated pregnancy associated with very severe disease in the fetus, in that fetus, there was viral protein seen in the brain, there was viral RNA as well, and a complete Zika genome was identified as well through deep sequencing analysis. And this was virtually identical to the other strains that have been circulating in Brazil. And there was also evidence of infection and injury in the neurons and tissue damage in the brain and the spinal cord. Next slide. So let me review what I think are the main unanswered questions that we have. How does in utero transmission occur, and why is Zika virus different than other flaviviruses? So unlike Zika virus, we've really not heard about microcephaly or other congenital malformations with dengue or West Nile virus, even though for dengue, certainly we have hundreds of millions of infections per year. So why is Zika virus different? I will remind you that there are other viruses, and Dr. Britt will talk about this later, but even the Flaviviridae family members have been shown to cause in utero infection. For example, before direct antiviral therapy, hepatitis C virus in approximately 4 to 5% of individuals could be shown to cause in utero infection. And the risk factor for this was high viral load in the blood. And the possibility is that Zika virus, like some of the other viruses, can infect cells in the layers in between the mother and the fetus, which then would allow or facilitate transmission across into the fetus and then to infect the fetus, presumably in cells in the brain, although we don't know that either yet in terms of where the virus actually goes. And is it the immune response, or is it the virus directly that's causing the injury? Next slide. A second unanswered question, and this was raised also before, is does pre-existing dengue immunity affect Zika virus disease immunity? We have no evidence for this at this present time, but this really has not been studied. Dengue virus, as Richard Kuhn showed, has significant homology at the E protein level to Zika. We know that antibodies are cross-reactive, and the question is whether the anti-dengue antibodies would cross-react with Zika and actually make things worse for Zika virus by allowing increased infection in myeloid cells. We don't know this yet, but it is conceivable. Certainly Brazil has a high population of people that are dengue immune, and one idea is in addition to virologic factors, there could be host factors associated with prior dengue immunity that could make Zika worse in some individuals. Reciprocally, it is possible that Zika virus immunity could affect dengue virus infection down the road. And these are 2 questions that we're going to have to study that we just don't have any answers right now on. Next slide. And then the third major area is what is the basis for Guillain-Barré syndrome that seems to be associated with Zika virus. And so there's, in the studies from Micronesia and now more recently from Brazil, there seems to be about a 20 to 25-fold increased risk of Guillain-Barré, although the data still is not totally clear. We do know that Guillain-Barré is a heterogeneous disease, and that it targets the myelin sheath around nerves and damages the myelin sheath, and patients who can get either paralysis, weakness, or paresthesias or tingling in their arms and their legs. What would be possible mechanisms? There could be immunopathology, so the antibody or T-cell responses that you generate to the virus could actually mimic a host protein. It could be that the virus has changed in its sequence and therefore now targets cells that are around neurons or near neurons, or there could be some other association that we just don't understand related to prior immunity to dengue. So really this is an area we need to study as well. Next slide. So this is my last slide, and I just sort of use it as a summary. And on the left side of the slide in the green is basically what we know. which is that there are Aedes mosquitoes that are the primary transmission points. We know the virus goes into the skin. It probably infects cells within the skin. We think dendritic cells might be a chance based on some preliminary data that's been published. Then you get viremia. This leads to the clinical syndrome, which occurs probably in 1 in 5 based on the numbers that we see. And ultimately, most people go on to protective immunity. However, in the blue area on the right is the questions that are really unanswered. Well, how do we get transplacental transmission, and how does this lead to congenital malformations? The issue of sexual transmission was raised. Why is Zika particularly unique among the flaviviruses? Does this lead to a persistent reservoir, as we have seen with other viruses, such as Ebola virus? What is the role of antibodies that are pre-existing from dengue, and what is the role of Zika antibodies perhaps on dengue disease reciprocally? Will this affect vaccine responses now that we're actually moving into deployment of dengue vaccines in the field? And what is the effect on Guillain-Barré syndrome, and how does that actually occur? And I'll stop there. Thank you very much.